Hi guys, welcome to Producing DNA Fragments. You need to be able to explain how complementary DNA is made using reverse transcriptase and explain how restriction endonucleases are used to cut DNA into fragments. So in terms of the specification, we're looking at the fragments of DNA production. So we will be looking at the processes that can lead to the production of the recombinant DNA, and there are 5 in total. So we've got isolation, we've got insertion, transformation, identification, and the growth. So for today, it's isolation. So how we can isolate the DNA fragments that have the gene of the protein that we are interested at. So to isolate the gene, we can use 3 methods. We can use reverse transcriptase. So it's an enzyme that's going to let us produce DNA from mRNA. So mRNA will act as a template. We can use restriction endonuclease enzyme, which can cut DNA. So those are 2 main differences between those enzymes. One is using mRNA to produce DNA, and the other one cuts DNA in 2 manners to produce blunt ends and sticky ends. And the final way of producing the gene, so a length of DNA, it's using the gene machine. So let's have a look how we can use reverse transcriptase first. So we are going to be looking at 2 different enzymes involved in this process. So first enzyme, reverse transcriptase, that will produce single-stranded DNA, cDNA from mRNA, and the second enzyme, DNA polymerase, which will then produce double-stranded DNA from cDNA. So how does it work? As an example, we are going to use the B cell from the pancreas, which codes for the insulin. Okay, so this gene will be, this mRNA will be isolated and will act as a template. So that's our single-stranded mRNA, which acts as a template. Reverse transcriptase enzyme then will come along and will be involved in the process of synthesis of complementary single-stranded DNA, which we will call cDNA. Okay, so this is our enzyme that comes along and produces single-stranded DNA, cDNA. The cDNA is then isolated by hydrolysis from mRNA. Okay, so that's the single-stranded DNA. So another enzyme that we've mentioned before, DNA polymerase, now comes along and produces a double-stranded DNA. So this is now a DNA, a gene, which codes for the insulin. Right, so that's the whole process. Second idea of isolating a gene, it's using restriction endonucleases. So those are the enzymes that can cut DNA. So we can cut DNA at a specific basis. We call them palindromic sequences or recognition sequences. So we can get 2 results of this cutting. We can get blunt ends. So that looks like this. So there are straightforward chopping portion of DNA, or we can get the sticky ends, and sticky ends will leave that complementary portion here that can then be used and stick together with another DNA from the other source. So have a look at the difference between blunt and sticky ends. So those are our blunt ends, so leaving the portions of DNA straight, and sticky ends, which we're cutting at the palindromic sequence. As you can see from the picture here, the palindromic sequence is really interesting, because if we read that sequence from 3' to 5' and from 5' to 3', okay, so here from 3' to 5' on one of the strands, the sequence is exactly the same as the sequence on the opposite from 3 to 5' on the second strand. So those are our palindromic sequences, the recognition sequences, where the enzyme can cut DNA, producing sticky ends. Right, so quickly how this is possible, let's make some notes. So sections of DNA has those palindromic sequences, which we've seen that they are read the same in opposite directions. Restriction endonuclease can recognize those specific sequences, can cut DNA different. Of course, restriction endonuclease will then cut a different recognition sequence because of the differences in the shape. So they need to be complementary to enzyme's active site. So if recognition sequences are present at either site of DNA you want, you can use restriction endonuclease to separate it from the rest of DNA, and this DNA sample can then be incubated with specific restriction endonuclease, which cuts the DNA fragment out through the process of hydrolysis. Then we will at the end get sticky ends, so those unpaired bases, and sticky ends can be used, what we've mentioned before, to be bind annual to another fragment of DNA from the other source. And that other fragment has to be cut with the same restriction endonuclease. Right, finally we've got a gene machine. So gene machine is a computer-based program that DNA fragments can be synthesized from scratch without the need for a pre-existing DNA template. So we just use the database to produce specific DNA fragments. And what are the advantages of this and how does it take place? So we only need the amino acid sequence of the protein that we are interested in. That will work out complementary DNA triplets. That sequence will be then fed into the computer. Complementary nucleotides will be added, those are called oligonucleotides, to assemble this specific gene. The advantage of using gene machine is the fact that we will not get any introns. So no introns in this process. Right, so that's everything for the production of DNA fragments. So we had reverse transcriptase, restriction endonuclease, and gene machine. See you later.